Hello friends, today we are going to discuss about tetrad analysis. Some of the lower eukaryotes, example unicellular algae and fungi, are used in the mapping studies or genetic mapping. These spend most of the part of the life cycle in haploid state. Among them, the most useful in genetic mapping is sac fungi or ascomycetes. This is because it has unique style of sexual reproduction. Fungi may be multicellular or unicellular and their cells are haploid. That means they have only one set of chromosome and these reproduces asexually. But these may reproduce sexually. Example, sac fungi or ascomycetes. In the sac fungi or ascomycetes, two haploid cells fuses and produces diploid zygote. Then chromosomal replication takes place, which is followed by zygotic meiosis, both meiosis 1 and meiosis 2, and four haploid cells are produced known as the spores, or these are also known as tetrad. Then these four haploid cells undergoes mitosis and produces 8 haploid cells known as octad. Both the tetrad and octad are contained within a sac-like structure known as ascus. So now let us discuss what is tetrad analysis. In the tetrad analysis, each product of the meiosis can be cultured separately, and then these are used to study their phenotypes, and on the basis of the phenotypes their genotypes are inferred or deduced, or the conclusion of the genotype is made. So we saw that after the fusion diploid zygotes are produced and then it is followed by zygotic meiosis, both meiosis 1 and meiosis 2, and in some cases mitosis also takes place, as a result of which spores are produced, and these spores are contained in sac-like structure known as ascus. So now we will discuss how these spores are arranged. Their arrangement varies from species to species. Here we will discuss about two types of the arrangement. One is the ordered or linear arrangement of tetrad or octad, and other is the unordered tetrad or octad. In ordered or linear tetrad and octad, tight ascus is present, that is, the sac-like structure is narrow. Hence, it prevents spores from randomly moving. So, tetrad or octad are linearly arranged. Example, Neurospora crassa. In ordered tetrad or octad, ascus is not tight. It provides enough space, as a result of which tetrads and octads of spores move randomly, and hence these are not linearly or orderly arranged, but these are unordered. Example, Saccharomyces cerevisiae. We will focus on ordered tetrad analysis. Let us discuss about the ordered or linear arrangement of spores in Neurospora crassa. Two haploid cells, 1n, that means these have only one set of the chromosome. For convenience, we have taken only one chromosome. Fuses, diploid zygote is produced, then chromosomal replication takes place. Now each chromosome has two chromatids that are attached to the centromere. Meiosis 1 takes place, the two chromosomes separate. This is followed by meiosis 2. Each sister chromatids of the chromosome separates and we get four haploid cells known as the spores that are contained in a sac-like structure. It is also known as tetrad. It is then followed by mitosis and we get 8 haploid cells which are present in the sac-like structure or ascus. It is also known as octad. Ordered tetrad analysis. It is used to map the distance or determine the distance between a gene and the centromere. This is the gene and this is the centromere. Then the distance between the gene and the centromere is determined, or it is used to determine the recombination frequency between a gene and its centromere. You can understand recombination frequency in my video on genetic mapping. The link is given in the description box. Two cases are possible that depends upon presence or absence of the crossing over between the gene and its centromere. Let us see first case where the crossing over is absent. So we have seen that haploid cells fuses and diploid zygote is produced, then the chromosomal replication takes place. So these are the two homologous chromosome. One has capital A allele and the other has small a allele. Then zygotic meiosis takes place, meiosis 1 followed by meiosis 2. After the meiosis 1, separation of the capital A and small a allele takes place. This is then followed by meiosis 2. Sister chromatids of the chromosome separates and we get 4 haploid cells that are linearly arranged, also known as tetrad. This is then followed by mitosis and we get 8 haploid cells that are linearly arranged. This is also known as octad. Here the 4 haploid cells containing capital A allele are adjacent to the 4 haploid cells containing small a allele. So the ratio of the haploid cell containing the capital A allele and small a allele is 4:4. It is also known as first division segregation or FDS because the separation or the segregation of the alleles capital A and small a takes place in the first meiotic division. Now the second case, crossing over is present. So there are two possibilities depending upon the two chromatids between which crossing over takes place. Here the crossing over taking place between these two chromatids, this part is exchanged with this part. After meiosis 2, these segregate and we get 4 haploid cell containing capital A, small a, capital N, small a alleles respectively. After mitosis, 8 haploid cells are produced that are arranged linearly, which is also known as octad. These are present as 2 haploid cells containing capital A allele, 2 haploid cells containing the small a allele, which is crossover type, and then again 2 haploid cells which has the capital A allele. These are also crossover type. Then again 2 haploid cells containing small a and small a allele which are non-crossover type. So the ratio is 2:2:2:2. So there is deviation from the 4:4 ratio as seen in the first division segregation. Now the second possibility, crossing over is taking place between this chromatid and this chromatid. So exchange of parts takes place between these two chromatids. And after meiosis 2, these segregate and we get capital A, which is non-crossover type, small a, which is crossover type, small a, non-crossover type, and again capital A, which is crossover type. After mitosis, 8 haploid cells are obtained, which are known as octad, and these are linearly arranged, and the ratio is 2 haploid cells containing capital A alleles, 4 haploid cells containing small a allele, and again 2 haploid cells containing capital A allele. So the ratio is 2:4:2. This is also deviation from 4:4 ratio that is seen in the first division segregation. In the first case, we have seen the segregation of the capital A and small a allele takes place in first meiotic division. But here the segregation of this capital A and small a allele takes place only in the second meiotic division. So it is also known as second division segregation, or in short SDS. Similarly, here also segregation of capital A and small a allele takes place in meiosis II. Now let us see calculation of map distance. Percentage of ASCII with SDS pattern for a gene is used to map the genes with respect to its centromere, or the percentage of ASCII with SDS pattern is used to determine the map distance between the gene and its centromere. Percentage of ASCII with SDS pattern is used because pattern of SDS is a result of crossing over. Crossover separates a gene from its centromere only when it occurs in the region between the centromere and that gene. This is the gene and this is the centromere. So this gene capital A is separated from its centromere only when crossover occur in the region between this gene and the centromere. So here crossover is taking place in this region, so this gene A is separated from its centromere. So the probability of getting the SDS pattern 2:2:2:2 or 2:4:2, it depends upon the distance between the gene which is taken into consideration and its centromere. That is, if the distance between the gene of interest and centromere is such that the crossover takes place in the region between these, then we will get the SDS pattern, either 2:2:2:2 or 2:4:2. So the map distance is calculated as half into number of SDS ASCII by total number of ASCII into 100. We have taken here half. This is because in the SDS ASCII, among total spores, only half of the spores are the result of crossing over, or only half of the spores are of crossover types. If we see here, among the 8 spores, only 4, 1, 2, 3, 4 are of crossover type. Similarly here 1, 2, 3, 4, that is half of the spores are of crossover type. In this way, using this formula, we calculate the map distance between the gene and its centromere. We are going to discuss about unordered tetrad analysis. What is unordered tetrad? In this, the spores are randomly arranged. Ascus provides enough space so spores can move randomly. So these are unordered. Example is Saccharomyces cerevisiae. It is used to map genes in the dihybrid crosses. Dihybrid cross is a cross that involves 2 genes or 2 loci. Through this unordered tetrad analysis, we can determine if the 2 genes are linked or they assort independently. And if the 2 genes are linked, then we can determine the map distance between them. 3 patterns are possible in the tetrads when the 2 pairs of alleles segregate. Example, if there is a cross between capital AB and small ab allele, which is a dihybrid cross, then 3 types of the tetrads are possible. Let us discuss them one by one. Type 1. This is the parental combination. If no crossover occur, then the product of meiosis is the same as that of the parental type. Or if double crossover occur between 2 strands, then also we get the product of meiosis having the parental combination capital AB, small ab. So if we see this figure, there is double crossover taking place between these 2 strands. So we get capital A, capital B. And here also small a, this cross goes like this, and then small b. These will be same, capital AB, small ab. So here also the product of meiosis is capital AB, small ab, small ab. So these are same as that of the parental combination. Hence the tetrad is known as parental ditype, PD in short, because of their resemblance with parental combinations. Type 2 4-strand double crossover occur between the two genes. Here one crossover is taking place between this and this strand, and one is taking place between this and this strand. So it is known as a 4-strand double crossover. And the product which we get after meiosis is capital A, small b, small a, capital B. Here capital A, small b, small a, capital B. So the products are capital A, small b, capital A, small b, small a, capital B, small a, capital B. So there are two types of the combination, capital A, small b, and small a, capital B. And none of the combination resemble as parental type, capital A, capital B, and small a, small b. So this type of the tetrad is known as non-parental ditype. Type 3, when 2-strand single crossover or 3-strand double crossover takes place between the two genes or occur between the two genes, it results in the third type of the tetrad. Here if we see single crossover taking place between these two strands, and the product that we get is 2 having the parental combination capital AB, small ab, capital AB, small ab, and 2 having the combination capital A, small b, and small a, capital B. Small a, capital B, and capital A, small b. These are of non-parental type. If 3-strand double crossover takes place, here the double crossover is taking place between 1, 2, and 3-strand. So the product of meiosis is, one is capital AB, other we get capital A, small b, capital A, small b. Then we get small a, it will go this way, small b, small a, small b, and fourth is small a, capital B, small a, capital B. So here also we get 4 types of the product, capital A, capital B, and small a, small b, small a, capital B, capital A, small b. These two are of parental type and these two are of non-parental type. We get 50% of parental type and 50% of non-parental type. This type of the tetrad is known as tetra type, or in short TT, because here all the 4 possible genotypes are present in the tetrad, and hence it is known as the tetra type or TT. Now in order to determine the recombination frequency or map distance, we use this formula NPD, non-parental ditype plus half TT, tetra type, by total number of tetrads into 100. So this is all for today's video. If you like this video, please hit the like button, share it and subscribe my channel. Thank you.